Hello, welcome to Biology, the scientific study of life. Notice this figure, a grass seedling growing toward light. This figure represents light-induced etiolation greening of dark grown potatoes. A represents before exposure to light. A dark grown potato has tall, spindly stems and non-expanded leaves. Morphological adaptations that enable the shoots to penetrate the soil. The roots are short, but there is still a need for water absorption because a little water is lost by the shoots. B. Notice also the differences between both. After a week's exposure to natural daylight, the potato plant begins to resemble a typical plant with broad green leaves, short, sturdy stems, and long roots. This transformation begins with the reception of light by specific pigment phytochrome. This represents also a review of general model for signal transduction pathways. A hormone or another signal binding to a specific receptor stimulates the cell to produce relay molecules such as second messengers. These relay molecules trigger the cell's various responses to the original signal. In this diagram, the receptor is one of the surface of the target cell. In other cases, hormones enter cells and bind to specific receptors inside. Notice cell wall, cytoplasm, receptor, hormone or environmental stimulus, plasma membrane. Notice receptor, reception, and relay molecules, transduction, response, activation of the response. Notice that. More about this is an example of signal transduction in plants, the role of phytochrome in the de-etiolation greening response. Notice reception, transduction, response. Notice the steps also. Cell wall, light, plasma membrane, cytoplasm, phytochrome activated by light, and CGMP, second messenger produced. Notice also the light signal is detected by the phytochrome receptor, which then activates at least two signal transduction pathways. Notice this Step 1 pathway uses CGMP as a second messenger that activates a specific protein kinase. The other pathway involves an increase in cytoplasmic Ca to the power 2 positive levels, which activates another specific protein kinase. Notice also specific protein kinase 1 activated response and specific protein 2 kinase activated. Ca to the power 2 positive channel opened. Notice that response transcription factor 1 nuclease, transcription factor 2, and translation de-etiolation greening response. Both pathways lead to expression the genes for proteins that function in the de-etiolation greening response. Notice the inquiry. This is another figure represent inquiry. What part of the coleoptile senses light and how is the signal transmitted? Experiment. In 1880, Charles Darwin and his son Francis designed an experiment to determine what part of the coleoptile senses light. In 1913, Peter Boysen-Jensen conducted an experiment to determine how the signal for phototropism is transmitted. Result: control light, illuminated side of coleoptile, shaded. Notice closer look, shaded side of coleoptile, Notice also Darwin and Darwin 1880. Notice that light, tip removed, tip covered by opaque cap. Notice also tip covered by a transparent cap, and notice tip base covered by opaque shield. Notice the Wojnsen 1913 light, tip separated by a gelatin block, and notice also tip separated by mica. Notice also the conclusion. In the Darwin's experiment, a phototropic response occurred only when light could reach the tip of coleoptile. Notice, therefore, they concluded that only the tip senses light. Wojnsen observed that a phototropic response occurred if the tip was separated by the permeable barrier gelatin, but not if separated by impermeable solid barrier, a mineral called mica. This results suggested that the signal is light activated mobile chemical. Another inquiry: does asymmetric distribution of a growth promoting chemical cause a coleoptile to grow toward the light? Experiment. In 1926, Fritz Wein's experiment identified how a growth promoting chemical causes a coleoptile to grow toward light. He placed coleoptiles in the dark and removed their tips, putting some tips on agar blocks that he predicted would absorb the chemical. On a control coleoptile, he placed a block that lacked chemical. On others, he placed blocks containing the chemical either centered on top of the coleoptile to distribute the chemical evenly, or offset to increase the concentration on one side. Notice that. Results: the coleoptile growth rate if the chemical was distributed evenly. If the chemical was distributed unevenly, the coleoptile curved away from the side with the block, as if growing toward light, even though it was grown in the dark. The conclusion: notice that offset blocks cause curvature, and control agar block lacking chemically has no effect. Control agar block with chemical stimulates growth, and excised tip placed on agar block, growth promoting chemical diffuses into agar block. Notice also the conclusion: when concluded that a coleoptile curved toward light because its dark side had a higher concentration of the growth promoting chemical, which he named auxin. Now, an overview of the plant hormones. Hormone auxin embryo were produced or found in a plant. Embryo of seed, meristems of apical buds, young leaves. Major functions: stimulate stem elongation, low concentration only, root growth, differentiation and branching, regulates development of fruit, enhances apical dominance, functions in photorobism and gravitorpism, promotes xylem differentiation, retards leaf apicogen. Cytokinins were produced or found in plants, synthesized in roots, and transported to other organs. Major functions: affect root growth and differentiation, stimulate cell division and growth, stimulate germination, delay syncytin. Notice also gibberellins were produced or found in a plant. Meristems of apical buds and roots, young leaves, embryo. Major functions: promote seed and bud germination, stem elongation and leaf growth, stimulate flowering and development of fruit, affect root growth and differentiation. Brasinosteroids were produced or found in plants, seeds, fruit, shoots, leaves, and floral buds. Major functions: inhibit root growth, recharge leaf apicogen, promote xylem differentiation. Abscisic acid were produced or found in plants, leaves, stems, roots, green fruit. Major function: inhibits growth, closes stomata during water stress, promotes seed dormancy. Ethylene were produced or found in plant tissues or ripening fruit, nuts of stems, aging leaves and flowers. Major functions: promote fruit ripening, opposites some auxin effects, promotes or inhibits growth and development of fruits, leaves and flowers depending on species. Inquiry: what causes polar movement of auxin from shoot tip to base? Experiment. To investigate how auxin is transported unidirectionally, researchers designed an experiment to identify the location of the auxin transport protein. They used a greenish-yellow fluorescent molecule to label antibodies that bind to the auxin transport protein. Then they applied the antibodies to longitudinally sectioned Arabidopsis stems. Notice the result. The left micrograph shows that the auxin transport protein is not found in all tissues of the stem, but only in the xylem branchium. In the right micrograph, a higher magnification reveals that the auxin transport protein is primarily localized at the basal end of the cells. Notice that. Conclusion: epidermis, cortex, folium, and xylem pitch. Notice that cell, the basal end of cell. Notice that. Conclusion: the results support the hypothesis that concentration of the auxin transport protein at the basal ends of cells is responsible for polar transport of auxin. This figure represents cell elongation in response to auxin at the acid growth hypothesis. Notice these represents cross-linking cell wall, polysaccharide, microfibril, and cell enzymes, expansion, cell wall, plasma membrane, cytoplasm, ATP. Notice when auxin increases the activity of a protein bumps to, notice H positive, the cell wall becomes more acidic. Notice that. Notice 3 wedge-shaped expansions activated by low pH separated cellulose microfibrils from cross-linking polysaccharides. The exposed cross-linking polysaccharides are now more accessible to cell wall enzymes. 4. The enzymatic cleaving of the cross-linking polysaccharides allows the microfibrils to slide. The extensibility of the cell wall is increased. Turgor causes the cell to expand. Notice that with the cellulose loosened, the cell can elongate. Plasma membrane, nucleus, cytoplasm, and vacuole. Notice cell wall and notice also elongation. Back or to cytokinins, epical dominance, the inhibition of growth of auxiliary buds, possibly influenced by auxin from the epical bud, favors elongation of the shoot's main axis Cytokinins, which are transported upward from roots, may counter auxin, stimulating the growth of auxiliary buds. The response being the removal of the apical buds from the same plant enabled lateral branches to grow. Notice stems after removal apical bud and lateral branches. Notice also auxiliary buds